Bismillah ar-Rahman ar-Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. In this video lecture, we are going to talk about eukaryotic flagella and cilia. So let me write. Flagella and cilia. Okay. So first of all, both flagella and cilia, both flagella and cilia inside the eukaryotic cell, it is very important for the movement of the cell. When the cell want to move from one place to another place, so it is possible by this flagella and cilia. How the flagella and cilia will be look like and how they are different from each other. Now let's suppose if this is a cell of eukaryotic cell, okay, and let's suppose it contain a nucleus, because all of you better know eukaryotic cell or those cells which have a well developed nucleus. Now this is the nucleus portion, and this now here means I'm talking about the difference between means the cilia and flagella. Now flagella are the long tail, okay, and if we talked about the cilia, so cilia are the thin hair like structure that are cover whole cell, as you are watching. Now this is flagella and this is cilia. Both are used for what? Both are used for the cell movement, okay. So when the cell move from one place to another place, it is possible by this cilia and flagella as well. Now this flagella, if we see the flagella and cilia, both act as a whip, okay. Whenever they beat, means whenever this flagella beat, so due to this beat the cell will move, okay. So let's suppose the flagella beat means whenever they are whenever they are performing their function, whenever the flagella are moving in sometimes they are moving in undulating, flagella are showing undulating fashions. Sometimes they are moving from tip to base and sometimes they are moving from base to tip. Due to this way the cell will move. So we call that what? We call that beat movement, and we also call that undulating fashion. Flagella and cilia both are differ from each other. Why? Because as I told you that flagella are the long tail of the eukaryotic cell and also present in a prokaryotic cell, and cilia are the thin hair like structure that cover whole cells. Okay, cilia, if we see, it's it's very means very small, and they having very small length as compared to this flagella. But on the other hand, both are similar to each other as well. Why they are similar to each other? They are similar because both are used for the function of the movement inside the cell. Both are used for the function. That's why we can say that it is similar to each other. One thing more, it is also a tail, and these cilia are also acting like a tail. So that's why we can say that it is means also what? Similar to each other. One thing more that I must tell you about the cilia, that cilia is act like a propel. Propel are specially used inside a board to push. Propel will be look like in this shape. Let me draw the diagram. And this propel, I hope you know about this propel. Propel is actually used inside the board. It is used in the board to pull the board or push the board to forward in the forward direction. Okay, and this propel will create what? And it will create what? It will create ore. Due to ore, the board will be move. And just like inside the eukaryotic cell, the cilia is also acting like a propel. Due to this way, the cell will move in a forward direction. Now, if we see the length of the cilia, so the length of the cilia will be 5 to 20 meter long. Okay, so it will be 5 to 20 meter long. Now, on the other hand, if we see the length of this flagella, so it will be 100 to 200 long. So this will be the length of flagella and cilia. This flagella can, as I told you, that flagella are showing the undulating fashions. What does it mean? It means that sometimes the flagella will generate a planar waves, and sometimes they will generate a helical waves. Okay, what does it mean? It means that, let's suppose if this is a cell, let me draw another cell here. Let's suppose if this is a cell, okay, then what actually happen? Now this is the flagella. So the flagella will sometime produce a helical wave, means they will produce a helical waves. As you are watching, this is the helical. Sometime they will what? They will create a planar waves, or generate a planar wave, just like that. Now this upper one planar and this lower one is the helical waves. Due to this helical and planar, we call that cell is undulating, means this flagella is showing undulating fashions. Due to this undulating fashion, the cell will move from one place to another place. Now one thing more, this, the flagella movement is possible from base to tip. Okay, when the flagella are showing their when the flagella are showing their movement from base to tip or from tip to base, then the cell will move from one place to another place. That's very important thing. Now one thing that I must tell you, when the movement of this flagella is from base to this tip, then what actually happen during this movement? During this movement, the cell will what? The cell will push. Okay, the cell will be what? The cell will be push. But when the flagella movement is from tip, means from this tip portion to this base portion, then what actually happen? The cell will be pulled. Okay, so this is push and this will be pull. Let me once again tell you, when the flagella show the movement from base to tip, then the cell will be pushed. Okay, and when the flagella movement from tip to base, then what actually happen? The cell will be pull. Okay, so it's very important things. Such pulling and pushing phenomena will be occur inside the water. Okay, so it's very important. Now if we focus on this flagella, then one thing I want to tell you about. Let's suppose this is the cell. Okay, then what actually happen if we assume this flagella? So it's very important thing that I must tell you, that sometime the flagella contain what? Sometime the flagella contain a hair-like structures. A thin hair-like structure. As I am drawing, this is a thin hair-like what? This is a thin hair-like structure. And this thin hair-like structure we call that filamar filament. Okay, now this hairs are called what? Filamar filaments. Now these filamar filaments are what? They are very thick. These hairs are very thick, stiffer and strong, and they are not bend. So it's very strong. Now we also call that what? We also call that mastigonemes. Mastigonemes. So we call that what? We call this here mastigonemes. Now when the flagella have these hairs like structure, we call this flagella what? We call this flagella tensile flagella. What we call that? We call that tensile flagella. Okay. On the other hand, when the flagella don't have this mastigonemes, then we call this flagella a necked flagella. Okay, what we call it? We call, let's suppose this one, they don't have what? They don't have this filamar filament. Then we call that what? We call that necked flagella. Necked flagella. When the cell have a tensile flagella, then it will be, cell will be what? The cell will be pull. The cell will be pull. Why? Because such phenomena, because during this structure, what actually happen? The movement will be, the flagella movement will be tip to base. Now let me once again tell you that what actually happen in this case, when a cell having the tensile flagella, so then this cell will be pulled. Okay, this cell will be what? This cell will be pulled. Now why? Because such flagella have a movement from tip to base. Okay, so that's why this cell will be pulled instead of pushing. Here one thing I forget to mention, that this next flagella, we also call that what? We also call it whiplash flagella. Whiplash flagella. Okay. On the other hand, if we discuss the cilia, so as I told you that cilia are what? Cilia are acting as a propel. Okay. Now cilia are also used. Means the cilia function are also used inside the fluid or inside the water. This cilia is act as a propelling, as I told you. Now what does it mean? It means that it's just like in boat, propel are used to create oars. Due to this oars, the boat will be move in a forward direction or in a backward direction. Just like in this case, the cilia are also used as a propel. Due to this way, it will also create an oar inside the fluid or inside the water, due to which the cell will be pull or push backward or forward. This is how the cilia function. Let's assume this flagella in more detail. Now if we see this the flagella, so one thing more that I must tell you about this flagella. So let's suppose this is the flagella. Okay, as all of you know, it is a long cylinder tube. Now this is a flagella. Inside this flagella, it contains more tubules. Let's suppose it contains more tubules. Each tubules is a pair. Each tubules are connected with each other in a pair forms. And each tubules will be in a pair form, and it will be connected with a central pair tubules. Now there are a central pair tubules, and all these inside the flagella, these tubules or cylinder, they have a diameter. If we see, so the diameter will be 0.2 meter in a diameter, and we call that what? We call that axoneme. We call that what? We call that axoneme. Now why we call that axoneme? Because these tubules are 9 pairs tubules. Each tubules are present but in a pair form, and all these tubules are 9 tubules, and each 9 pairs tubules are connected with this central pairs tubules. Okay, all these connected with this central pairs tubules. Now if we further assume it, and if we see the top view of this picture, now let's see this picture more in detail, and let's focus this picture on a top view. Now if we see the top view, so what actually happened? Let's suppose this is the tubules circular form. Now this is this one portion, and this is the top view form. Top view. Now what actually happened? As I told you that it is connected 9 pairs tubules. 9 pair what? They means each flagella contain 9 pair tubules, which we call that axoneme. So let me draw here. These are what? These are the tubules. 9 pair tubules. 2, 3, 4, 5, 6, 7, 8, and one more. This is 9. Okay. So 9 tubules, and each 9 tubules are connected with what? It is further connected with the central pair of tubules. Now one thing more, that this central microtubules are it is covered by a central sheet. So it is what? It is covered by a central sheet. Now this is what? This is the central sheet. Now each microtubules, as you are watching, each pair of microtubules are connected with each other by an Axene link. Now this is what? This is the Axene link. Okay, so it is connected with the Axene link. Now one thing more, this one is called, this first tubule we called it subtubule A. Sorry, subtubule B. Subtubule B. And this second one we call that subtubule A. So one thing that I must tell you, this Axene link, this is what we call that Axene link. Now this Axene link are actually arises from the subtubule A and connected with the subtubule B. This is very important. And if we see all these tubules is connected to each other, okay, all these tubules is further connected with this central microtubules. Now this is the central microtubules, but it will be connected with a radial spoke. This is what, this is the central this is the central microtubules. These are the central microtubules. Okay, now this is, as you are watching this layer, we call this what? We call this central sheet. Central sheet. So this all this pair of the tubules, means all this 9 pair of the tubules are connected with this central microtubules with the help of radial spoke. Now this one means this one area, as you are watching, we call that what? We call that radial spoke. Radial spoke. So one thing more that I must tell you, that there will be a head portion, as you are watching. These are what? These are the head portion. So this head portion is called what? This head portion, we call that spoke head. Okay, so we call that spoke head. And these two are what? These two are called microtubules. Tubules. Now one thing more that I must tell you, each microtubules is similar. Each microtubules means each pairs of the microtubules which are present in the flagella, it is similar, just like the microtubules that are present in the cytoplasm. If you remember, we talked about the tubules, microtubules and cytoskeleton. If you remember, we also talked about this. If you missed that video, so go back to my channel, it is present. Now each tubules is similar to what? To the microtubules that are present in the cytoplasm. All these patron, all these, this pairs and this central microtubules and this 9 pairs tubules, we call that what? We also call that 9-2 microtubules. 9-2 microtubules patron. Now why we call that 9-2 microtubules patron? Because each tubules are 9 pairs, first thing, and the next thing is that each 9 pairs tubules are connected with this central microtubules pairs, and it is also 2. So that's why we call that 9-2 microtubules patron. All these we call that 9-2 microtubules patron, and we also call that what? Axonemes. So that's all about the axonemes, that how it will be look like. One thing more that I forget to mention, these Nexine link, as you are watching, these Nexine links are connected with what? It is connected with a pair of arm, as I am drawing. These are what? These are the pair of arm. This arms, these arms are 2 arms. Now these arms are 2 arms. The first arm we call that outer Dianine arm. Why? Because it is an outside, it is present an outside. And if we see this one arm as present at the inside, we call that what? We call that inner Dianine arm. Okay, so each 2 arm arises from this Nexine link, and these 2 arm, the first arm we call it outer because it is present outside of the Nexine link, and the one we call that inner, this inner Dianine arm, because it is usually present inside area. Now each microtubules, as I told you, it is similar to the cytoplasm. So each microtubules is composed of actins protein and contractile protein as well. Okay. So now one thing more that I must tell you about the cytoplasm and cilia. Keep one thing in your mind, that the base area of the cytoplasm and cilia will be present in the cytoplasm area. Okay. Now as you are watching, this is what? This is the base area. So it is usually present in this cytoplasm area. Just like if we see this cilia, so this cilia is also present Their base area is also present in their cytoplasm area. One thing more, each cilia and flagella will be growing when the microtubules are connected to their tips. Let's suppose there are the microtubules which are similar. So if the microtubules are connected to here, so then the flagella length will be elongated. Similarly, if the microtubules are connected with the cilia, so then similarly the cilia length will be also increased. So it's very important. If on the tip area the microtubules are connected, so then both cilia and flagella will be what? Elongated. Now one thing that I forget to mention, that these arms are called Danine arms. One we call that inner Danine arm and other we call that outer Danine arm. Now each Danine arm, if we see the length of each Danine arm is what? The length of each Danine arm is 15 nanometer long. Okay, so it will be 15 nanometer what? Long. And each 15 nanometer it is long, and each Danine arm is actually made up of Danine proteins. It is what? It is made up of Danine protein. As it is made up of Danine protein, that's why we call that Danine arms. Now if we see if we see the speed of this flagella movement, so the speed of the flagella movement will be 260 meter per second. What does it mean? It means when they move at this rate speed, so it means that they will cross almost 40 cells per second. Okay, so it will cross 40 cells per second. So whenever, let me tell you once again, whenever this flagella is showing the speed, means if the flagella speed is 260 meter per second, so it means that they will cross almost 40 cells in a length. So they will cross 40 cells in a length per second. So we have different kinds of examples of the microorganisms that have different kind of speed of the flagella. Okay, so the first microorganisms we have what? We have Euglena flagellate. Euglena flagellate. Okay, so Euglena flagellate, if we see, so it has 170 meter speed. And what does it mean? It means that they will cross almost 30 cells. And one thing more, we have another bacteria, we have another microorganisms, we call that Euglena gracilis. Euglena gracilia. Euglena gracilis will be, moving at a speed of this 170 meter per second. So Euglena gracilias and Euglena flagellate, both eukaryotic microorganisms will be moved at a speed of what? 170 meter per second. And it means that they can cross 30 cells per second. Now another example we have ciliated protozoa. Let me write ciliated protozoa. Ciliated protozoa paramecium caudatum. So ciliated protozoa paramecium caudatum, if we talked about this microorganism, so this microorganism swim at a speed of 270, means 2700 meter per second. So they will be moved at that rate of speed. What does it mean? It means that they will cross almost 100 of cells per second. Such kinds of speed and such kinds of cells, flagella function and movement and everything are not observed in a prokaryotic cell or in a bacterial cell, because bacteria and prokaryotic cells have not all these structure and have not all these speeds. It is very difference. There are a big difference from each other. If you miss the prokaryotic flagella, so I also discussed the prokaryotic flagella in detail, so go back to my channel and just visit it. Okay, so that's all about the eukaryotic flagella and cilia. I hope you understand about this video lecture. If you like this video, so make sure to hit the like button, share this video, and don't forget to subscribe the channel. Subscribe the channel to get more interesting videos like that. Thank you.